Okay, and the first one we'll talk about today is bacterial enzyme. So different bacteria can produce different types of enzymes, and these enzymes can help them to infect the host. This include like enzyme related to tissue modulation, including like tissue degrading. And those enzymes can help the bacteria dissemination in host tissue. Here is a list of different enzymes in this category, like collagenase, streptokinase, and hyaluronidase. Those are different enzymes, and also coagulase. Coagulase is not a tissue degrading enzyme, but it can cause the clotting of serum. So it turns fibronectin to fibrin, and fibrin is not soluble, which are long protein fibers. So that's a role of coagulase. The function of coagulase is that it can block the attack of immune cells. So it coagulates the fibrin so that immune cells cannot get access to the pathogen, to the bacteria. And collagenase, that's an enzyme which can degrade collagens. And we know that collagen is also a protein fiber which is rich in connective tissues. And by degrading the collagen, the bacteria can make gaps in tissues so that they can spread within the tissues. So that's the collagenase. And there are also other enzymes, like the one at the bottom, that's IgA1 protease. And we know that IgA1 is the antibody, the IgA type 1 antibody. And this protease can split the antibody. You know, the immune system made this antibody to fight against infection, but the bacteria somehow evolved the ability to produce this protease, which can specifically degrade the antibody. It's an important secretory. IgA1 is an important secretory antibody on mucosal surfaces, and this protease can inactivate the antibody activity, so that makes themselves safe. So these are the different types of enzymes which are related to virulence. Another virulence factor is the anti-phagocytic factors. On our immunology classes, we learned that some of the cells can phagocytize particles, including bacteria and virus, so they can take up those particles and kill those particles inside the cell. This process we call phagocytosis. And for phagocytosis, there is one way that the phagocytes express Fc receptor on their surface. And this Fc receptor can bind to the Fc fragment of antibodies. This is an antibody. So that this antibody recognizes a bacterium and then bring the bacterium to phagocyte. The interaction between the receptor and antibody Fc region will take the bacteria into the phagocyte so that the phagocyte can kill the bacteria inside the cell. But the protein A produced by Staphylococcus, that's the species called Staphylococcus aureus, which can produce protein A, and protein A can bind to the Fc region of antibodies, the red one. Bind to the Fc region so that it blocks the interaction between the receptor and the Fc region, so that it inhibits phagocytosis and carry out the anti-phagocytic effect. So this is a different virulence factor. The last one we'll talk about is bacterial biofilms. When we culture the bacteria in the lab, usually the bacteria will grow in single cell form. So they form a suspension or form a colony, but they are single cells. But in the real situation, in the natural environment, either like in the environment, like in the river, in the water, or in the human body, once the bacteria or microorganisms colonize in these natural conditions, usually they are not living by single cells. They are not living in single cell state. Instead, they stick together to form a special structure, a slime structure. So a lot of different types of microorganisms, including bacteria, fungi, virus, and parasites, and even like amoeba, single cell organism, they live together. They secrete extracellular polymers like peptide or polysaccharide, and those extracellular substances will stick them together to form a slime-like structure, and this slime-like structure is called biofilms. And microorganisms, including bacteria, living in biofilms are more resistant to antimicrobial agents like antibiotics. They are more resistant to chemicals like bleach, and they are more resistant to liquid flow. For example, in the river, if they are in single cell state, the water flow will take them away, wash them away. But if they form a biofilm at the bottom of the river, and that will make them stick to the surface, and the flow of the river cannot take them away. And also it happens in our body, like in the different tracts. The biofilm can help them to stick there well. Here are some examples of biofilms. This is like the famous picture of human teeth. And you can see the yellow color of the human teeth. That's because there is a layer of biofilm, a layer of microorganisms sticking to the surface of the teeth, which gives them color. The color is because they can produce some pigment, and they can cause inflammation on that site. And now in the hospital, some people have their joint replaced. This is the artificial joint. This is not a real bone, because in the real bone bones there are immune system, there are vessels and immune cells, and which can prevent infection. But in this artificial joint, it's just metal or plastic, and there is no immune components, so that there is no molecules or cells to inhibit the growth of microorganisms on the surface. So usually microorganisms can stick to the surface and to form biofilms, which cause chronic infection. That happens a lot. And also in the blood vessel, the biofilms can form there. Okay, so right now for the modern medicine, to fight against biofilm is one of the major topic people are working on. Okay, I'd like to show you a short video on bacterial biofilm, and it shows us how the biofilms form.
Such as pipes and basin walls. Flowing bacteria in the bacteria produce an extracellular polymeric substance or slime that holds the growing biofilm surfaces, walls, films are not chill biofilm microorganisms develop in the bulk film underneath biofilm to a variety of problems in water systems. Corrosion.
Okay. And more set against action. Because we learned this and each branches have their own properties. First, for the innate immunity, this is the first rapidly. They lack the immunologic memory, so immunity does not form memory cells. But sorry, can you hear me? No? It seems that the network, some issue happened to the network. Can you hear me now? Hold on, please. Okay. Okay. All right, thank you. I think it's stuck somewhere when we played the video. What something happened at that time. Okay, all right. Let's continue our lecture. Just ignore the video. That video introduces how biofilms form. And we know that the biofilm is one of the virulence factor, and it's not easy to treat a biofilm because microorganisms living there are more resistant to antimicrobial agents, and they are more resistant to like liquid flow. Now we are switching to another point, that's a host defense against bacterial infection. Okay. All right. And we know that the immune system contains two branches. One is the innate immunity, the other is the adaptive immunity. The innate immunity is the first line of defense. And this immunity is not specific. So it does not fight against the microorganisms specifically. So any microorganisms invading the system will be cleared by innate immunity, and they can respond rapidly. But it lacks immunologic memory. This is a traditional belief that innate immunity does not form memory cells. But in recent years, advances have shown that some innate immunity cells, like NK cells, can actually form memory. That's the very recent results from research. It's not in our textbook yet. Composition of innate immunity contains different layers, like a physiologic barrier. That means our skin or mucous membrane. Those barriers can block the invading of pathogens. So they are the actually first line of defense. And there are also professional compositions, including professional immune cells like phagocytes, including granulocytes. This is mentioning like the neutrophils, or macrophages, monocytes. All of them can take bacterial particles into the cell and kill them in the cell. So they are phagocytes. And also natural killer cells, NK cells. And there are also molecules, immune molecules, that we call the humoral factors, including complements, which can help to lyse the pathogens, and the lysozyme. Lysozyme is, we introduced this lysozyme before. It's an enzyme produced by the host system, which can degrade the cell wall of bacteria. That's the lysozyme. This is the innate immunity. And here is an example of phagocytes, and we can assume that this is a macrophage. The macrophage can take up the microorganisms or other particles, the small particle, into a vacuum and take it into the cell. And in the cell, we call it phagosome, because it's formed through phagocytosis. This phagosome will fuse with another vesicle called lysosome. Lysosome contains a lot of digestive enzymes, including like protease, DNAs, a lot of enzymes. And two vesicles will fuse together. And when it fuses, the enzymes will degrade the bacteria. Will degrade the bacteria intracellularly, so that the bacteria is killed intracellularly. This is the function of phagocytosis, to kill the bacteria. But on the other end of this process is that once the bacteria is killed intracellularly, this macrophage can also present the antigen to adaptive immune cells, right? So that's what we learned in immunology. Okay, another branch of immunology is adaptive immunity. Adaptive immunity is a second defense system, and this system can react to pathogens specifically. So, and also it can form immunologic memory and can form memory cells like memory T cells, memory B cells. And there are two different types of reaction. One is antibody mediated. Antibody are secreted protein molecules. The antibody mediated immunity we call humoral immunity. Another type of immunity is cell-mediated immunity. And this we call cellular immunity, like CD8 positive T cells. which can kill infected cells. So this is cellular immunity. Okay, so this is a second branch of immune system, adaptive immunity. This area actually it contains a lot of material, what we learned in detail in immunology class. Here I'd like to show you a very nice video. This video describes different roles, different players of the immune system in fighting bacterial infection. So it describes the story when our body is infected by some bacteria, and how the immune system, how the different players of the immune system join the battle to clear out the bacterial infection. That includes the macrophages, T cells, B cells, neutrophils, and also cytokines, chemokines, antibodies. A lot of players together. They join the battle one by one, and finally they can clear out the bacterial infection. Okay, I really like this video. It's very nice.
Every second of your life, you are under attack. Billions of bacteria, viruses, and fungi are trying to make you their home. So our bodies have developed a super complex little army with guards,
Can you
soldiers,
hear the audio?
intelligence, weapons factories, and communicators.
Okay, let's continue.
To protect you from, well, dying. For this video, let's assume the immune system has 12 different jobs. For example, kill enemies, communicate, etc. And it has 21 different cells and 2 protein forces. These cells have up to 4 different jobs. Let's assign them. Here are the interactions. Now, let's make this understandable. First of all, let's add colors to the jobs. Now, let's illustrate the cells. The central color represents the main job of the cell, while the surrounding ones represent secondary duties. Now the immune system looks like this. Now the interactions. Isn't this complexity just awesome? For this video, we will only talk about these cells and ignore the rest. So what happens in the case of an infection? It's a beautiful day when suddenly a wild rusty nail appears and you cut yourself. The first barrier of the immune system is breached, your skin. Nearby bacteria seize on the opportunity and enter your wound. They start using up the body's resources and double their numbers about every 20 minutes. At first they fly under the radar, but when a certain bacteria population is reached, they change their behavior and start to damage the body by changing the environment around them. The immune system has to stop them as fast as possible. First of all, your guard cells, known as macrophages, intervene. They are huge cells that guard every border region of the body. Most of the time, they alone can suffocate an attack because they can devour up to 100 intruders each. They swallow the intruder whole and trap it inside a membrane. Then the enemy gets broken down by enzymes and is killed. On top of that, they cause inflammation by ordering the blood vessels to release water into the battlefield, so fighting becomes easier. You notice this as a very mild swelling. When the macrophages fight for too long, they call in heavy backup by releasing messenger proteins that communicate location and urgency. Neutrophils leave their patrol routes in the blood and move to the battlefield. The neutrophils fight so furiously that they kill healthy cells in the process. On top of that, they generate barriers that trap and kill the bacteria. They are indeed so deadly that they evolved to commit suicide after 5 days to prevent them from causing too much damage. If this is not enough to stop the invasion, the brain of the immune system kicks in. The dendritic cell gets active. It reacts to the signals of the soldiers and starts collecting samples from the enemies. They rip them into pieces and present the parts on their outer layer. Now, the dendritic cell makes a crucial decision. Should they call for antivirus forces that eradicate infected body cells or an army of bacteria killers? In this case, antibacteria forces are necessary. It then travels to the closest lymph node in about a day. Here, billions of helper and killer T cells are waiting to be activated. When T cells are born, they go through a difficult and complicated training process, and only a quarter survives. The surviving cells are equipped with a specific setup, and the dendritic cell is on its way looking for a helper T cell with a setup that's just right. It's looking for a helper T cell that can bind to the parts of the intruders which the dendritic cell has presented on its membrane. When it finally finds one, a chain reaction takes place. The helper T cell is activated. It quickly duplicates thousands of times. Some become memory T cells that stay in the lymph node and will make you practically immune against this enemy. Some travel to the field of battle to help out. And the third group goes on to travel to the center of the lymph node to activate a very powerful weapons factory. Like the T cells, they're born with a specific setup. And when a B cell and a T cell with the same setup meet, hell breaks loose. The B cell duplicates rapidly and starts producing millions of little weapons. They work so hard that they would literally die from exhaustion very fast. Here, helper T cells play another important role. They stimulate the hard-working factories and tell them, "Don't die yet, we still need you, keep going." This also ensures that the factories die if the infection is over, so the body doesn't waste energy or hurt itself. But what is produced by the B cells? You've heard of them, of course, antibodies. Little proteins that are engineered to bind to the surface of the specific intruder. There are even different kinds of antibodies that have slightly different jobs. The helper T cells tell the plasma cells which type is needed the most in this particular invasion. Millions of them flood the blood and saturate the body. Meanwhile, at the site of infection, the situation is getting dire. The intruders have multiplied in number and start hurting the body. Guard and attack cells fight hard, but also die in the process. Helper T cells support them by ordering them to be more aggressive and to stay alive longer. But without help, they can't overwhelm the bacteria. But now the second line of defense arrives. Billions of antibodies flood the battlefield and disable lots of the intruders, rendering them helpless or killing them in the process. They also stun the bacteria and make them an easy target. Their back is built to connect to killer cells, so they can connect and kill the enemy more easily. Macrophages are especially good at gnoming up the bacteria which antibodies have attached to. Now, the balance shifts. In a team effort, the infection is wiped out. At this point, millions of body cells have already died. No big deal, the losses are quickly replenished. Most immune cells are now useless, and without the constant signals, they commit suicide, so as not to waste any resources. But some stay behind, the memory cells. If this enemy is encountered ever again in the future, they will be ready for it and probably kill it before you even notice. This was a very simplified explanation of parts of the immune system at work. Can you imagine how complex this system is? Even at this level, when we ignore so many players and all the chemistry. Life is awfully complicated, but if we take the time to understand it, we always encounter endless wonders and great beauty.
Okay, this is really a nice video. It summarizes how the immune system responds to bacterial infection and contains a lot of information. And you guys can keep this video. If you want, you can find the video on YouTube. Okay. Now we have a little bit information left to talk about in this topic. That's the sources of the infection. Talking about the sources of infection, we are talking about where the pathogens come from. Is it from outside, from the environment, from another person, or is it on yourself, which causes the infection at some time point? So based on this, we can classify the sources of infection as exogenous infection. So that's the pathogen is from outside, either from some patients or some carriers. Another person who healthy, but another person who is healthy, but he carries some pathogen, and that pathogen can transmit to another person and cause infection. So they are carriers, or might be from animals, Like we talk about the Black Death, the plague, which is an infection of Yersinia. And usually there is a reservoir in the environment. Some animals carry the bacteria of Yersinia, and the Yersinia can somehow spread from the animal field to human and cause Black Death. Okay, so this is the exogenous infection. The other one is endogenous infection. So that's infection from inside. We do have a lot of bacteria on our body, like bacteria on our skin, on our mucus, or in our digestive tract. And most of them form a normal flora. They don't cause disease usually. But at special conditions, for example, our immune system is compromised, and we are stressed, and at that time point, some of them can rise up and cause trouble to our body, and this is endogenous infection. This slide includes a lot of information talking about different sources of infection and also the spread of infection. Here on our class, we won't talk about the detail in this figure, but you can spend more time in this figure and figure out more information. The sources and spread of infection. And also we need to learn the pathways of bacterial entrance. Different bacteria enter the host in different ways. Some can infect our body through direct contact, like touches, kisses, which can transmit bacteria from one person to another. And some of them are a sexual contact, and some of the bacterial STD disease. So bacterial STD, like gonorrhea, which can be transmitted through sexual contact. And some bacteria can enter our body through inhalation. So that's a respiration, including mycobacterium. So that's the pathogen causing TB and other microorganisms. Some of them can enter our body through ingestion. So when you drink or eat something which is not fresh, which is contaminated by the pathogen, and they can enter our body through the food or water or fluid, and then can cause infection. And this includes different bacteria like Salmonella, Shigella, Vibrio, and also Clostridium, and they can get into our body through ingestion, and also animal vectors like Yersinia pestis, okay, which can be transmitted through like ticks or other animal vectors. Okay, so this is the pathway of bacterial entrance. Different types of bacterial infection include, we talk about the virulence, we talk about the pathogenicity, whether they can cause disease or not. And we also talk about their virulence. That's the degree of pathogenicity, how much disease they can cause, what level of disease they can cause. Is it mild or is it severe? Is it critical? And based on this, we can classify bacterial infection into different groups, like inapparent infection. So in this group, there's no obvious symptoms or few symptoms. It's not detectable, and there is no clinical symptoms. And apparent infection. Apparent infection means that the symptoms are obvious and can be detected. Some infections are setting up quickly, so once the bacteria get into our body, they can cause acute infection, so the sudden onset and rapid progression, and often with severe symptoms like cholera. The cholera enters our body and can set up infection within hours. So that's acute infection. Some are chronic infection, which means that after entry of the pathogen, it takes a long time and shows a delayed onset and slow progression. So this is a chronic infection. Or carrier state. We talk about the carrier persons. They carry the pathogen, but they don't show any symptoms. They are not ill. They are not sick, but they carry the pathogen, and they can also spread the bacteria to others. So they are carrier state. Okay, so generally we have covered a major point in this long topic, the bacterial infection and immunity. We actually talk about these glossaries or these terms during our lecture, like infection, pathogen, non-pathogen, normal flora, opportunistic pathogen, and also bacteremia, septicemia. Some of them we did cover, but some of them we didn't. I would like to leave these glossaries for you guys to learn by yourself, and you need to understand the definition of each term, and this will help you to understand our textbook. Okay, so I will leave these 2 slides for yourself, and you need to learn independently by yourself. We won't cover it here on our lecture. Okay, so that's all for this topic, this bacterial infection and immunity. We'll have a break, maybe 10 minutes, and after break we'll start another topic on bacterial infection diagnosis and prevention. Okay, and we'll be back in 10 minutes.